At this time, I'd like to welcome our 4 new Breakthrough Prize laureates to the stage. This year's recipient of the Breakthrough Prize in Fundamental Physics, Saul Perlmutter from UC-Berkeley and Lawrence Berkeley National Laboratory. 2 of this year's winners of the Breakthrough Prize in Life Sciences, Jennifer Doudna, also from UC-Berkeley, and Emmanuelle Charpentier from the Helmholtz Center for Infectious Disease. And finally, one of this year's winners in the Breakthrough Prize in Mathematics, Terence from UCLA. Please welcome our guests. Thank you.
Good to see you. Nice to see you. Hi, good to see you. Alrighty, this is great. If you thought those last guys were interesting, this is going to really knock your socks off. So now we're getting the rubber's meeting the road here. These are the people who are really at the intellectual frontier, and I'm gonna go through each person here and ask them to tell us a little bit about what they do, but we're not going to get bogged down too much in a lot of the explanation which you heard last night in that terrific evening that we had. But we're going to try to get to some of the fun stuff and some of the motivations. I mean, science is kind of to the general public, science can be kind of intimidating. It can be hard to understand. It's kind of elitist maybe. But when you talk to people who are doing it, you realize that it's just exciting, and it's a supreme act of human creativity, and it has all the drama and serendipity of any human story. So. Now that I've raised your expectations, here we go. Saul, can you tell us? You were working in cosmology, and you had, there was a bit of serendipity involved in your measurements of supernovas. You were trying to figure out trying to show that the universe acceleration was slowing, but something went wrong.
So, well, my colleagues, Brian Schmidt and Adam Rees and I, our 2 teams were doing what we thought was going to be for me actually, one of the most fun projects I could imagine, because we were measuring what we thought would be the slowing rate of the expansion of the universe. Gravity would slow down the expansion. And the reason I thought it was such a great project was because whatever answer we got, would just be an amazing philosophical result. If it turned out that it was slowing some, but not enough to ever stop slowing, then we knew at that point that the universe was infinite and it would expand forever. And I mean, measuring just the brightness of these little dots of light, these supernova explosions, and finding out the universe is infinite, I thought was great. Alternatively, we could have found out that the universe was slowing enough to come to a halt and then someday collapse, in which case we could have been measuring that the universe was going to come to an end. And I thought, measuring these little dots of light and being able to walk around with a sign saying that the universe is coming to an end just before the millennium, I mean, it seemed like you couldn't go wrong. And yet when we actually made the measurement, we found out something that was none of the above. It turned out that it was, you threw the apple in the air and you waited for it to come down, and it didn't. It just blasted off. And the universe apparently is speeding up in its expansion. And of course, even for a scientist, even better than an amazing answer to a question that you thought you had is discovering a whole new question. And so that was the surprise.
I mean, I love stories like that because it really shows it's completely the opposite of the stereotype generally held by scientists about science, that it's finding answers, and it's really finding crazy questions that you didn't know. So then you were motivated purely by personal gain while you're doing this, and
purely it was.
Yeah. Okay. That was a joke. Thank you. And next we have
if you find out that the real estate market, if the universe really is going to always expand, then you know which way to put your.
There we go. Okay. All right, Jennifer Doudna and Emmanuelle Charpentier. You guys have a great story, which, by the way, is the cover story in this issue of Scientific American, which I'm shamelessly promoting right now. It's available right outside. You have a great story because you were doing basic research. Would you consider it basic research in how bacteria fight viral infections, right? Can you tell us a little bit about how that went? Well, I don't know which one of you might want to start or the other. I don't know which went first, but you started doing this work, and then you started to you got the idea to collaborate.
Yes, so actually I was not initially interested in understanding how bacteria fight their own viruses. I was interested in this question at the beginning of my career, and then I moved a little bit away from this field.
Oh, I see.
To really try to understand the mechanisms that bacterial pathogens are using to cause diseases in humans.
Right.
Always with a focus on really fundamental mechanisms with the hope to find targets for new anti-infectives, new therapeutics. And it's just by focusing on one of these regulatory pathways that I came to link one certain molecule of RNA with a CRISPR-Cas system. And so I was back to my former love, which actually I had left also because this was a kind of research that was considered to not be really that fashionable. You know, studying bacteria and how they interact with bacteriophages, the viruses of bacteria. It's considered a little bit as old macrobiology, even though a lot of the tools that we are using in molecular biology nowadays, all the tools that we are using for manipulating genes are coming from fundamental research, trying to understand how the bacteria defend themselves against invading DNA. But it was not that fashionable, and I think for me, it's a very good example of how do you say, really very, very important to not especially follow the fashion or follow the dogma and just follow your curiosity and maybe by deciphering a pathway you will find something interesting. And then you will combine two pathways that maybe were not meant to interact. And then, and then you can just follow and discover things that you would not have expected to discover.
So this one molecule led you back. So how did you start working? How did you start working together?
I think it's a great example of a project that came about really through serendipity. I mean, I think we are people working, traditionally had been working in very different areas of science. We might not have even met each other. But, you know, I had gotten interested in what I thought at the time was a very esoteric project, you know, to try to understand how bacteria fight the flu using very small pieces of RNA stolen from viruses. And it seemed like a fascinating, you know, just biological phenomenon. And so I had, you know, very, very little bit of grant support from the National Science Foundation and actually also from the Gates Foundation to do this work. And so it started with just one person in my lab investigating this. And eventually, you know, it was so interesting that more and more students started to join the project. And we actually got invited, or I got invited to an American Society of Microbiology meeting, something I would have never attended. I'm really not a microbiologist at all. But that's where I met Emmanuelle, and we realized in talking that first of all, we really liked each other, which was great. That's important for collaboration. And also, we realized that our laboratories had complementary expertise. My lab is really a lab that's always been interested in molecules and how they work, and what they do, and particularly RNA molecules. And her lab, as she said, you know, had an interest in a particular pathogen that had this CRISPR pathway. And so we got together to understand the fundamental mechanism of a protein called Cas9 that was clearly important somehow in the function of this immune system in strep pyogenes that Emmanuelle's lab was studying. And that led to, you know, we had no idea where that fundamental research would go, but as soon as we figured out how this protein worked, we realized this could be an incredibly exciting tool for genome engineering.
I love stories like that. I love it because it involves, not just following some logical plan of research, but it involves a lot of serendipity and curiosity and meeting people that you happen to get along with. And there was also another connection in your lab, right? Because you were separated geographically.
Yes, so me, I'm using a lot of Skyping because I don't want to. Skype does not sponsor my research, but we are using a lot of Skype.
But Skype sponsors everything.
I have 2 laboratories and I have moved a lot around the world, so I'm collaborating very easily.
But there were Polish speakers?
Exactly. So actually, my student, all the projects started with my student when we discovered the components that would be involved in this pathway. And then when when I contacted Jennifer, actually, she chose to ask Martin Jinek, a postdoc from Czech Republic, to join the project. So that was my student, Krzysztof Szyliński from Poland. And actually, they can understand one another. It's like, so they could speak. He was speaking Polish, and Martin was speaking Czech.
They had never met each other, right? But I came back from this conference, and I said to Martin, who was a postdoc in my lab, there's this really exciting project. Would you like to work on it? And he said yes. And so he started talking with Christoph in Emmanuelle's lab across, 6,000 miles and using Skype. And they pretty quickly figured out they had both grown up in a sort of Eastern Europe on opposite sides of the Polish border. And they actually both spoke the same dialect of Polish, of all the serendipitous things, right? And so they actually communicated often in their native language. And so this was just a really fascinating connection. We couldn't have planned it, but it actually made the social aspects of the science go very smoothly.
And I think what is really important also in the collaboration is generosity. I know from my part, my student is very generous, and I think it's the same thing for Martin. When you collaborate, you should not be afraid to just tell all your ideas and
share your data.
To just go ahead.
Generosity.
And this was very important.
That's good. That's great.
And it's always what scientists are a bit scared of, because you have tough competitions, and you're always scared that you're going to be scooped by someone if you say something. Right. But I think it's very important because. You want to go ahead with your ideas, with a pathway, and you need also to see whether someone is going to catch and respond, and so it's important for.
So, and of course, this collaboration led to technology for gene editing that is, from everything we hear, is going to make some pretty big, incredible changes in genetics and make genetics far easier to do, genetic manipulation far easier to do, and far cheaper, and will lead to a lot of good things. Okay, Terry Tao is a mathematician, and mathematics is a tough one. I have read your biography, and I still can't really say what you do. And I think, it's kind of the opposite in some ways to some of these more pragmatic sciences that we've just been talking about. But so I know that you started at a very young age. You showed a math propensity to do math at a very young age. What is it like to have that gift, and have we, and when I say we, I mean society, have we made it easy for people like you? Because you have a gift, and it's a gift that we all benefit from. And I'm just wondering what that journey's been like, and what we can do, how people have helped you on it.
Okay, yeah. So I've loved mathematics for as long as I can remember. One of my earliest memories was as a child, I think 3 years old. My grandmother was washing the windows, and I was demanding that she put the detergent on the windows in the shape of numbers. And I always liked, my parents told me that if they wanted to keep me quiet in the evening, they would give me a math workbook. And I think I always liked things with very clear rules, things were black and white. I was terrible at the humanities as a child. I think I had an English assignment once to describe what was going on in my house. And I just went to each room and wrote down a list of everything, all the objects that were in every room. So, I always had a very literal mind, and I always liked sort of very abstract things, so I naturally gravitated to mathematics. I was very lucky to have so much guidance. I think my primary school headmaster and high school teachers and then later professors in college were very accommodating. They allowed for a special schedule where I would go to school, say in primary school with people my own age, and then I would take math classes 4 or 5 years ahead of my age group in high school, and then when I was in high school I'd teach classes in college. And poor mom had to drive me around every day like 5 times a day. But yeah, I think I was perhaps lucky that when I was growing up there was not much precedent for this sort of thing, and there was a lot of leeway for the individual administrators to actually bend the rules a little bit for me.
Yeah. So how do you figure out what to work on?
Well, I have. So a lot of it's sort of following your nose. There's questions you're interested in, and when you solve a problem, naturally other problems just naturally appear afterwards. But a lot of it is serendipity, as we saw. Most of my favorite collaborations are from things I just didn't expect. But I'll be talking to a colleague, and they would mention a problem which turns out this is connected just to something I was interested in. One of my favorite collaborations was with an applied mathematician, Emmanuelle. They were working on, he and another colleague, Justin Romberg, were working on a way to try to improve the image reconstruction for MRI, to take fewer measurements of human body and then try to get as sharp image as possible. And the current imaging methods needed maybe 2 or 3 minutes of imaging to get a decent image. So they took a test image, a theoretical test image, took fewer measurements. The standard reconstruction didn't work very well. So they tried a new reconstruction method, hoping to get a slight improvement. And what they ended up doing when they ran their simulation for the first time, the reconstructed image looked almost perfectly like the original, using only maybe a tenth of the measurements that we thought were necessary, but they couldn't explain why. And so Emmanuelle came to me and said, I've got this funny result. And he was able to translate this problem, which was very practical, into a problem about a very mathematical problem about random matrices, actually. And I looked at this and said, this can't be right. You have so little data, and you're getting such a perfect reconstruction. So I actually spent the night trying to disprove, trying to show that what he was saying couldn't possibly work. But then I found actually that there was an explanation why this was happening, and so we started working together. And and now actually we have algorithms that can speed up MRIs by a factor of 5 or 10. It's the only practical thing I've ever done. But it was completely serendipitous. Yeah.
That's funny. People don't. I don't normally think of mathematics as being collaborative. But how does that happen? Is it do you go to conferences? Do you Skype people? Do you seek out advice by people working in some area that you need advice on?
All of the above. Yeah. So it's changed a lot culturally. I think maybe 50 years ago it was definitely a very individual, competitive environment. People raced to be the first to prove a theorem, were very jealous of this. If they had partial progress, they would keep it to themselves.
Right.
And you would wait until your papers were published before sharing them too much. And it was, but now it's changed. I think the nature of mathematics has changed. I mean, the problems we face are much more interdisciplinary. Naturally, we need more collaboration. But plus, collaboration is so much easier now with the internet. Most of my co-authors are in other universities or overseas, and it's just much more fun actually to collaborate.
Fun, that word keeps coming up.
Oh yeah.
So this is the global brain at work, I guess, right?
Yeah. No, I really feel like I'm part of an international community. Yeah.
So I'm gonna ask everyone this, but we might as well keep with you. So what are you working on now? What's your next thing? What's your current obsession?
It keeps changing. I don't know often what I'm be working on next year or the year after. I have plans that they always get derailed by events. One thing I'm working on now actually is to study a set of equations called the Navier-Stokes equations that govern the evolution of fluids. And it's a long-standing open question in mathematics whether these equations they can ever develop solutions which are smooth initially but develop singularities. Like maybe could they create fluids which approach infinite velocity in finite time, which can't happen physically. And what that would mean is that these equations are not actually a good model for actual physics. And so this has been a long-standing question for some time. But I think now there is a chance to show that this is actually possible, that if you have a carefully constructed solution of fluid, you can actually make a sort of a self-replicating machine. So the configuration of fluid which turns itself into a smaller version of itself, which turns itself into an even smaller, faster version of itself, and within a finite amount of time, it actually creates a singularity. But to do that, I have to make a machine just basically do mechanical engineering out of pure water, which I don't know how to do yet.
Okay. Anybody has any ideas, he'll be available after.
That's one long-term thing I'm looking at.
You know what the amazing thing is? I can actually kind of understand that. So thank you. You're very, very clear explanation. Saul, we haven't talked to you in a while. I want to ask you the same thing. You did this really great work, and so what are you doing now? What have you done for us lately?
So right after we had seen this surprising result that the universe is accelerating, the theorists went to town trying to explain what could possibly cause the universe to speed up in its expansion. Right. You know, gravity would tend to slow, you would expect. And in fact, I was noticing that there's been a theoretical paper published every 24 hours for the past 16 years on average, trying to explain this. And if you ask any of the theorists, do you stand by this theory? They'll cheerfully say to you, no, I'm just trying to expand the range of possibilities. Really, the ball's back in your court, the observers, the experimentalists. You've got to give us a little bit more of a clue to go on as to some of the properties of this, this perhaps dark energy, if that's what it is, or what else is going on in the universe. So we've been, the whole community now has really dived into this question of can we develop new techniques for measuring in much, much more detail than we did before, things like the history of the expansion of the universe. These different theories of dark energy, if that's what it is, predict very slightly different histories of the expansion. So we need to make that kind of measurement 20 times more precise than we did before. And that's going on now with work for, I've been involved in the supernova side of that story. And for example, just yesterday, or last night, I guess, we were expecting data in from the Hubble Space Telescope that would allow us to make the measurement much, much further away. The data didn't come. I was asking Adam, why we weren't getting our data yesterday. And then there's just a few other techniques on the table, because it's a very difficult thing to measure. Other colleagues here in the room have been working on techniques to measure the clumping of stuff in the universe, the actual material in the universe that forms the galaxies and the clusters of galaxies. And that clumping also can tell you about perhaps these different theories of why the acceleration. So those are the games that we're now playing.
So what does it involve to get to increase by 20 times the sensitivity of these measurements? I mean, do you do that by yourself, or do you have teams of engineers working on it, or what?
I mean, these are all, again, I mean, the projects that we did before were collaborations of 20 or 30 people. The next projects are collaborations of that size and larger. So part of the problem is, I have one collaboration now that's trying to calibrate the supernova much, much more precisely, so you can use them as these distance measuring yardsticks with much, much more detail than before. So that's a project, international project. France, Germany, are involved, Sweden. But there's another angle which is going to require even more people. We've been developing a space telescope that would be like the Hubble Space Telescope, but with a field of view 100 times bigger, several hundred times bigger. And that's obviously a project that will end up, needing the whole infrastructure of a space project. And so, that one we're already working with teams of 100 people.
So that's going to cost a lot of money. So what's the payoff here?
Well, so this is the classic issue of fundamental science, right? Which is one of the whole points of this prize, which is the fact that we do not understand why it is that over and over again, magically, when you learn something really deep about how the world works, somehow it makes us more capable. And I don't think anybody could have possibly guessed, for example, in my field, that if you began trying to study questions like Einstein's theory of general relativity asks about what happens when clocks travel near the speed of light, that would ever possibly be a practical something that you could sell, and yet it turns out that nowadays we rely on our GPS systems. All of us are, we probably are here today in this room literally because we followed our GPSs to.
I would still be driving around.
Precisely. And that depends crucially on things like the fact that general relativity gives you the corrections. You would miss, by dramatic, the plane would miss the runway if your GPS didn't use Einstein's theory of general relativity. But clearly, if you'd asked Einstein at that time, so why are you doing this? How's it possibly going to help anybody? He probably would not have. I'm sure he would not have been able to tell you what it would someday allow us to do.
So you wouldn't want him writing the grant application.
And similarly, I think to this day, when people ask us, what are we doing with dark energy, it's very hard to argue that this should be, why are we sponsoring this in the Department of Energy, in the United States? It's, clearly we don't really expect that tomorrow it's going to solve an energy crisis, even if 3/4 of the universe might turn out to be made out of dark energy. We don't think it's practical in that way, but we think that what you learn about the underlying physics of the universe may turn out someday to allow us to do something that we never could have done any other way.
Well, dark energy sounds like it'd make a great weapon, I would think.
Yeah, well, the dark side of the universe. It's
right. Okay. What about you guys? Are you now this technique that you devised? Are you engaged in refining it, working on it? Have you moved on? What are you up to?
Well, so in my lab, I would say really we're doing two things. One is trying to understand actually how it works. I think it's quite an amazing thing to think of a protein and a little piece of RNA that's able to seek out a sequence in the human genome and recognize it specifically and allow cells to make a change at that site. So we'd really want to understand the molecular process by which that happens. And actually Sam Sternberg, who's a graduate student in my lab, has been one of the people in my lab really driving forward that effort, and he's actually here today. And the other direction we're going in is, I think everybody appreciates this is going to be is already a very powerful technology for genome engineering in all sorts of different systems. And so one of the things that I did with a lot of help from our dean, Steve Martin, who's here, and others at Berkeley, Graham Fleming, of course, and Ed Penhote, who's also here, is to establish the Innovative Genomics Initiative, which is a joint project between UC-Berkeley and UCSF. We were fortunate to recruit Jacob Korn from Genentech to be our scientific director. He's also here today and was there last night. And the goal is really to look for opportunities not only to continue to do great basic science and sort of bringing together basic research at Berkeley with clinical researchers at UCSF, but also to figure out exciting ways to partner with companies. And so we've already been able to establish two very important relationships with companies. AstraZeneca is one of them, and we're looking for opportunities to move this forward in the future. And I think we heard earlier from our leadership that this is clearly an important direction for the future of research. And the question that was asked by Wendy, a student, this is, I think, one of the ways that we're going to fund basic research going forward is to look for ways to partner with companies and look for opportunities that, really bring together the best of what both have to offer.
Great. What about you, Emmanuelle?
Yes, so we are continuing also understanding the molecular mechanism of Cas9, focusing on the biochemistry, which was actually our initial focus. Try to understand a little bit the details of the biochemistry of the mechanism. And also because I moved to Germany relatively recently, so I have established a number of collaborations in the regenerative medicine field, and also involved in a biotech company and working together with pharma to develop further the technology for the purpose of using the technology to treat human diseases.
So you're really following this into in an applications phase?
Yes, partly.
Yeah, partly.
But otherwise, my lab is really focused mainly on other mechanisms, molecular mechanisms involved in the interaction between bacteria and the human host. And so we are looking at different pathways, and I really want to pursue this research because I just hope to make new interesting discoveries and to have the chance to hit in the future, interesting pathways. I'm not sure whether, I think it will be very challenging to hit a molecular pathway that can have so much applications as CRISPR-Cas9, but at least in the anti-infective field, I think it's very important to pursue a fundamental research
Right.
to try to.
So how has it changed your life to find something that has this vast practical benefit? Because there's now it sounds like that's become a whole big bandwagon now. And does that, so is there a tension between that and the basic research you still want to do, or?
Yeah, for me, I think it's, let's put it that way. When you do research, you never know what you're going to find, and you never know what kind of applications it will lead to. I just think that for me, it provides me confidence, and I think to my team as well, to just pursue the research and to be confident that sometimes it may take some years. And sometimes it's also important to maybe try to connect different aspects that are not logically to be connected, and that one can be successful and discover.
Well, success is very good for the confidence. That's good. So how do you feel about the way that CRISPR is perceived by the general public? There's been a lot written about it. Some people worrying about what the benefits, but also some of the potential misuses of it. Do you think about that? Does that affect you? Do you have any opinions? I'm asking both of you.
Well, I would say, for me, I think that's a very good point. One of the things that's happened is that this technology has taken off so quickly. I mean, certainly faster than I think either of us could have anticipated. That it means that it's kind of out in front of where regulatory agencies might be thinking, and even scientists. How do we use this safely and effectively? And so I think that's actually one of the goals of the IGI that we established between our campuses, is to think about this, sort of the bioethics around this. We have an upcoming meeting that includes very prominent people from around the country to come together and really have a day of brainstorming about this and thinking about it. And I don't think any of us have, or I certainly know I don't have the answers for that right now, but I think it's an important conversation to have. And I think it gets to your point about how CRISPR is being portrayed in the media, how people are learning about it. I feel that it's very important to communicate this science to a lay audience, to the public. And I think, I'm really grateful to Yuri Milner and the Breakthrough Prize folks for putting together the kinds of documentary little film clips that we saw last night and that will be shown broadcast in the future, because I think this is a great way to educate people about the science and maybe get them interested to look into it more, or at least to understand the basics of what it's doing.
Yeah. So I mean, so the downsides, the potential abuse is that it makes it possible to do things that now takes a big lab and lots of expertise. Perhaps that doesn't take quite as big a lab and quite as much expertise, and that has a potential for abuse. But a lot of technologies that we've greatly benefited from over the years have had this same. Have been two-edged swords as well, and we deal with them by discussing it and trying to get out ahead of it ethics-wise. So I think that's it's important to say that.
Yeah, for sure.
Okay, so we've got a few minutes and we've got microphones around. I thought maybe perhaps someone would like to ask a question. I can do this all day, but you guys are pretty smart. Do we have any hands? Shall I keep going? There's a microphone coming for you. And please tell us who you are.
I'm Suzanne Pfeffer, Stanford University School of Medicine. Just to be clear for the audience who don't use the Cas9 technology, so people really understand what it can do and that it's nothing to be scared of at all. This is an ability for a scientist in using a cell line that we study in the laboratory to make a change, to fix a genetic mutation that may cause a disease. The question that we're hearing about in the future is that the ability to take an embryo perhaps and correct a disease before the baby is born. Or, just to be very explicit, so there's nothing to be scared of today, nothing to fear the technology today, but it could be incredibly useful in the future to treat disease and also maybe even in a particular tissue. We already can use other kinds of technology in the eye to cure diseases of the eye, and it may be able to be used locally. So I just was hoping you could explain a little bit more clearly some of the bioethics questions that you just raised so that we shouldn't be concerned. This is powerful technology for the future that will help a lot of patients, we hope.
That's a great question.
I mean, I guess the point that I would make, Suzanne, and thanks for bringing that up, I think is that, what I think people need to appreciate is that it really is a very powerful technology. It really means that scientists now with, anybody in science who has training in molecular biology, we've had, even high school students come to the lab over the summer, and within a few weeks, they are editing human cells that are cultured in the laboratory, right? So there's that. So it's very simple to use and doesn't require a lot of expense or expertise. But it's also a very powerful technology. And so as you alluded to, I think the kinds of research experiments that you just described are fantastic, and many labs around the world are already doing this. I think the challenge going forward is thinking about what about engineering human embryos? Is that a direction that science should go, wants to go? And if yes, then what kinds of genetic mutations should be corrected? Should it be anything goes, or should there be some thoughtful approach to how we might do that? So that's what I would say. Yeah, it's not to be afraid of, but it's definitely something that it's very powerful, and I think that there needs to be widespread appreciation for that.
Yeah, and it's a technology that the biology and the medicine fields are going to profit a lot. It's changing a lot the face of biology because a number of scientists working on different model organisms, including human cells, were not really able to manipulate genes in a simple way. And now they can manipulate genes so they can really understand the functions of those genes. So this is after the area of sequencing all those genes and figuring out the genetic code, then what are the genes doing. So I think the technology is very transformative at this level. And then it's transformative in the medical field because beyond the fact that the technology itself can be used as a tool to treat human genetic disorders, it can really be extremely helpful to create these models of diseases that are extremely crucial for the pharma to understand how drugs are working, and also in terms of screening for new drugs. And it's also important for all the engineering of proteins, antibodies, and also in the ag bio field. So it's a very broad technology, and I think before to see the negative side, there are so many positive output that I think it's important to focus on those.
Okay, great. Right here, we have a microphone over there.
I'm Pitch Johnson, a local venture capitalist and a graduate of Palo Alto High School. But I wanted to ask a question that just came up in the previous discussion. The rewards in science, especially in science, but in all disciplines, are the deep drillers. You four are deep drillers in your subject, and you go down and you discover things. But the connecting the deep drilling, connecting things in one discipline to the next, has never been rewarded, at least highly, by academe. Do you see any signs of that happening? Do you anticipate any change so that the connectors of people of different parts of science and other disciplines are going to be rewarded or an important part of academe?
The connectors.
One place where I see some motion in that direction came up in the previous discussion is this area of one of the commonalities of many of the sciences now, and in fact, something Matthew describing is the data science areas. And we've recently started seeing attempts to get a chance for people to take advantage of the fact that across the whole campus at a university, for example, at Berkeley, there are people who are, that is with their real skill set in whatever subject domain they're in. They're doing the best in the data analysis side of it, and that they can learn from each other and work across the campus much more fluidly than people in other areas with other kinds of expertise. So at least at some of the campuses now, in the universities, you're beginning to see the desire to reward that and to find ways to give long-term positions, permanent faculty positions that would be able to bridge in that particular way, where previously people might have thought that, that person's not a physicist, they're a computer scientist, or that person's not a computer scientist, they're a physicist. Now there's now an interest in trying to find ways to support those interconnecting people who are able to bridge many fields.
Interesting.
But it's just beginning in some cases.
Yeah, I think that biology has already started a while ago to connect with other disciplines. I mean, biophysics, physics has been integrated in medicine. Physics is also integrated. All the mathematical models now for all the interactions, the molecular interaction, the new molecular interaction that are discovered. So I think there are already some interconnection, biocomputational analysis. So, but clearly it can be developed further.
It's certainly in mathematics, the culture has changed dramatically. I mean, it used to be that each field, subfield of mathematics was its own sort of silo of specialized knowledge, and they were almost proud, some people in the field would almost be proud that what they're doing is incomprehensible even to other mathematicians. But now, if they find that something they do is of interest to, say, a life scientist or a linguist or something, it's very exciting, and the culture has changed a lot. At UCLA where I work, we have a whole institute, the Institute for Pure and Applied Mathematics, which is devoted pretty much to fostering these connections, actually. We have these programs which are deliberately designed to span a wide spectrum of expertise, from pure math, applied math, and sciences, and even the social sciences. So we had, I think, a program on the mathematics of the heart, and so we had cardiologists, and we had biologists, and we had a whole spectrum of people. So I think the culture is really moving in the interdisciplinary direction.
Great, and I know a lot of university librarians and educators, publishers like our sister company Nature are working on ways of being the glue or the connective tissue between scientists who are collaborating across disciplines. Any other questions? Here we go.
Hello, my name is Libby Willis. I am a graduate student at UC-Berkeley, and this is directed a little bit towards Terence, although you kind of touched on this already. But even if you think of how this panel discussion has gone, what you have studied does seem pretty, unreachable or esoteric even for a lot of us here who are scientists. And mathematics often seems kind of scary to most people, especially younger kids. And so how do you foresee, reaching out to, younger people or, either college students or high school students who may be good at math, but, what you're studying just doesn't seem, attainable or even able to process. So how do you foresee reaching out and kind of spreading math knowledge and what you do?
Well, that's a great problem. I think, yes, it's true that my discipline has done a much worse job than the physicists and the life scientists for selling what we do and making it accessible to the public. Partly we can justify it because we work on things that are inherently more abstract. But we can certainly do more. I think as suggested earlier, we really should have much more of an emphasis. Every sort of as part of a full academic career, we should do more public outreach. In fact, so when I do public lectures, I often rather than talk about pure mathematics, I often I can talk about how mathematics is used in the sciences as being more accessible. The public lecture I give the most often actually is on the cosmic distance ladder, which I sell as a way of using mathematics to measure the universe. Yeah, because then I can leverage all the lovely pictures and data and wonderful things that
And because it's so practical.
well, it's more practical than most of what I do. So, okay, that's yeah, but that's so yeah, you have to be maybe a bit more creative in mathematics than in the sciences to sell what you do. But I think it can be done.
I guess I would just add to that so I'm obviously not a mathematician, but I have a 12-year-old son who is very, very interested in math, loves math. And what we've found, so he goes to public school in Berkeley, and he is sort of beyond, what is being done in his classroom right now, as are some of the other kids. And one of the great things that's happened, and I wonder if this is happening elsewhere too, but at Berkeley, several math professors got together and they actually started an evening math club. They call it Math Circle for kids. And this is incredibly popular. I would say there are probably hundreds of kids that come to this every Tuesday night at Berkeley. And these professors they teach these kids. They do it just for no pay, and they just come up with really fun, creative sort of little games that the kids do. And every time I pick my son up after Math Circle and I ask him, how was it tonight? And he's always really excited. He's like, Mom, tonight we learned about topology or, whatever, right? And it's really great to see. And so I think that through those kinds of outreach things, it doesn't cost a lot, and it's really a way to engage kids in the world of, in this case, mathematics, where they feel like it's fun and it's about problem solving and puzzles.
My son goes to a Math Circle at UCLA too, and it's very, very popular. I know there's a huge untapped demand for this. In fact we're scrambling to find enough postdocs and students to teach. Yeah, no, that is certainly a great outreach tool.
Well, this is great. I think we're running a little over here, so we're going to have to cut it short. But I want to thank you all for listening, and I want to especially thank our really wonderful panel of brilliant scientists who've to come out and talk with us about what they do. Thank you. Let's give them a hand.
So, first of all, thank you so much to the Breakthrough Prize Foundation for the incredible recognition provided to these wonderful scholars. Thank you, Fred, for your great job of moderating the two panels. We were delighted at Stanford to host this event, but it couldn't have happened without a really great partnership between the Breakthrough Foundation and UCSF and Berkeley and Stanford. And thank you to this wonderful audience for joining us today. You are welcome to stay for coffee and tea out in the Ford Gardens, as this room will be prepared for the Life Sciences Symposium, which starts at 11. You're also welcome to join the Mathematics Symposia, which will take place at Bechtel Conference Center, which is about a block and a half away that direction, or the Physics Symposia that will take place at the Karettaub Hall in Gunn-Sieper Building, which is a little bit around the corner. All three symposia begin at 11, and they run throughout the day until 5 o'clock, and box lunches will be provided at the symposia. A schedule of talks and directions to each symposium are available at your tables and at the check-in desk at the lobby. So once again, thank you all for joining us. It was a great morning.